Homo Sapiens. Who are we? Where do we come from? In Chapter 17, we are going to continue our quest to answer the question of how life began on Earth. In Chapter 16, we looked at some basic time frames for the beginning of life on Earth. The current evidence points to the possibility of life emerging on Earth roughly around the 3.8 billion year mark or possibly earlier. This provides us with a general idea of when life emerged on Earth. As we begin answering the question of how life emerged on Earth, we are going to need to cover a broad array of topics ranging from the Earth's evolving atmosphere to organic chemistry to the various scenarios hypothesizing the emergence of living organisms from non-living, complex organic molecules. Before we start digging into these more complex topics concerning how life began on Earth, let's take a moment to look at the classifications of life on Earth today. Life on Earth can be broadly classified into two categories, prokaryotes and eukaryotes. Prokaryotes are single-celled organisms which have no central membrane-bound cell nucleus, nor do they have any membrane-bound organelles. Compared to the eukaryotes, the prokaryotic cell structure is fairly simple. The DNA of the prokaryotes is suspended within the cell's cytoplasm in a single molecule called a nucleoid. Some of the prokaryotic DNA is also stored within the cell's cytoplasm in separate circular DNA structures called plasmids. Prokaryotes are strictly unicellular, but they can form communities which are found encased in a polymer matrix or slime referred to as a biofilm. Eukaryotes, on the other hand, are distinguished by their membrane-bound cell nucleus which contains the cell's DNA. Eukaryotes also have membrane-bound organelles such as mitochondria, which are specialized structures that perform specific functions necessary to the cell's survival. Eukaryotic cells are also much larger and more complex than prokaryotic cells. All multicellular life forms on Earth are eukaryotes. Humans, trees, and mushrooms are eukaryotes, as well as single-celled organisms such as paramecium, euglena, and didinium. The prokaryotes and eukaryotes fall under a three-domain classification system. The prokaryotes are classified under two domains, bacteria and archaea. The eukaryotes fall under the third domain, eukarya. The domain eukarya includes the kingdoms of Animalia, Plantae, Fungi, and Protista. Before we dig deeper into the classifications of life on Earth, let me digress a bit to define a few terms we will be using. The first term is phylogeny. A phylogeny is essentially the evolutionary history of a genetically related group of organisms. The phylogeny, or evolutionary history of a group of organisms, can be depicted as a branching tree-like diagram, which is essentially a genetic family tree. These branching tree diagrams are called phylogenetic trees. Phylogenetics is the study of the evolutionary relationships between groups of organisms based on their morphology or physical characteristics and their genetic makeup. With the advent of DNA sequencing, genetic relationships are increasingly relied upon to construct a phylogenetic tree. If we look at this diagram or phylogenetic tree of the three domains, we can get a sense of the evolutionary relationship between the three domains as well as the organisms within the domains. Each branching event on the tree represents a genetic split or speciation event within a population of organisms. The speciation event results in the divided population heading down separate evolutionary paths. The lower on the tree the branching or speciation event, the farther back in time it occurred. Looking at the phylogenetic tree of the three domains, we can see that the existence of the organisms in the domains can be traced back to a sin ancestor or last common ancestor. Understanding the nature or possible characteristics of this last common ancestor may help us answer the question of how life appeared on Earth. Let's take a moment to examine the nature of the organisms in the three domains of life on Earth. This comparison will give us some insight into their phylogeny, that is to say, their evolutionary history and genetic relationship. As we take a closer look at this phylogenetic tree of life on Earth, let's focus in on some of its more important aspects. This particular phylogenetic tree is based on the sequencing of ribosomal RNA or ribosomal ribonucleic acid, which is abbreviated rRNA. Ribosomes are highly complex structures found in all living cells, prokaryotic and eukaryotic. Ribosomes perform the function of protein synthesis within the cell. Ribosomes consist of two subunits that fit together, a small subunit and a large subunit. These two subunits work as one to translate messenger RNA into a polypeptide chain during protein synthesis. The instructions or recipes for protein synthesis are stored within an organism's DNA. Messenger RNA reads the protein recipe stored in the DNA, then transfers that recipe to the ribosome, which then converts the recipe into a chain of amino acids, forming a protein. Proteins themselves carry out a vast array of duties critical to the functioning of living organisms. Ribosomal RNA sequences are widely used for working out the evolutionary relationships between organisms due to their ancient origin and the fact that they are found in all living organisms. Let's also note the position of the syn ancestor, or last common ancestor, at the root of the phylogenetic tree. This represents the last common ancestor to the domains of bacteria and archaea. One important thing to note from the phylogenetic tree is that the early members of the domains of bacteria and archaea are the aquifex for bacteria and the core archaeota for the archaea. Both the aquifex and the core archaeota are thermophiles found living in hot springs or high-temperature hydrothermal environments. As both aquifex and core archaeota are descendants of the syn ancestor, as well as being rooted closely in time to the speciation event leading to the lines of bacteria and archaea, their thermophilic characteristics may be evidence that the syn ancestor itself was thermophilic in nature. This hints at the possibility that the earliest life forms may have thrived in a high-temperature hydrothermal environment. The first branching of the phylogenetic tree occurs with the genetic split in the population of the syn ancestor, which led to the rise of the domains bacteria and archaea. This indicates that at some point in the distant past, possibly before the 3.8 billion year mark, a genetic division occurred in the population of the syn ancestor, leading to the separate lines of the bacteria and the archaea. The bacteria and archaea share similarities in cell structure and metabolism, but they differ in the composition of their cell walls. Whereas bacteria have cell walls made of peptidoglycan, archaea have cell walls made of pseudopeptidoglycan. Another difference between bacteria and archaea is that no pathogenic, that is disease-causing archaea, have ever been identified, unlike the bacteria. Another item to note is that even though the bacteria and archaea both fall in the category of prokaryotes, the archaea appear to be more closely related to the eukaryotes than to bacteria. This is due to the fact that the archaea and eukaryotes have been found to share many important genes that are involved in DNA replication and repair, as well as DNA transcription and translation, genes which are not found in bacteria. Even though the archaea and the eukaryotes seem more closely related to each other than either are to bacteria, the actual origin of the eukaryotes is a bit of an enigma. The gene complement of the eukaryotes is an uneven mixture of genes which seem of an archaeal origin and those of a probable bacterial origin, as well as genes that are specific to the eukaryotes themselves. The question is, how did such a genetic chimera as a eukaryotic cell arise? The leading hypothesis on the origin of the eukaryotes is the endosymbiotic theory. Stated briefly, the endosymbiotic theory holds that an anaerobic prokaryote, unable to use oxygen for energy, lost its cell wall. The more flexible underlying cell membrane began to grow and fold in upon itself, leading to the formation of a cell nucleus and other internal membranes. This now primitive eukaryotic cell began to ingest prokaryotic cells as food. At some point, the eukaryotic cell failed to digest and engulf bacterium. The primitive eukaryotic cell thus commenced a mutually beneficial or symbiotic relationship with the prokaryotic bacterium, in which the eukaryotic cell provided protection and nutrients, and the prokaryotic bacterium provided additional energy to its eukaryotic host via the bacterium's respiratory cellular machinery. This symbiotic relationship became permanent over time as the bacterium lost genes needed for independent life. Organelles found in eukaryotic cells, such as mitochondria and chloroplasts, are hypothesized to have originated through this endosymbiotic process, whereupon a once free-living entity engulfed within the eukaryotic cell eventually becomes incorporated into the eukaryotic cell's biological processes. Once the eukaryotic cells appeared, they seem to have undergone a fairly rapid rise to complexity by evolutionary standards. The eukaryotic cell progression to the multicellular level marked the beginning of much more complex life forms. The earliest known fossil evidence of possible multicellular organisms has been dated to 2.1 billion years ago. This would put the origin of eukaryotic cells back before the 2 billion year mark, possibly as early as the 2.5 billion year mark. This puts the origin of eukaryotic cells within the time frame of the Great Oxidation Event, which occurred around the 2.4 billion year mark. The Great Oxidation Event marks the appearance of free oxygen in the Earth's atmosphere. Let's now flesh out a rough timeline of the emergence of life on Earth based on the information we have gathered from Chapter 16 and thus far in Chapter 17. The Earth forms around the 4.54 billion year mark. By the 4.4 billion year mark, evidence from the Jack Hill zircon crystals points to the Earth's crust having cooled and formed. By the 4.3 billion year mark, the Earth's hydrosphere has formed with the resultant oceans, rivers, and lakes. The Earth's surface is still highly volcanic and suffers numerous bombardments from comets and asteroids. Somewhere around the 4 billion year mark, we see the emergence of life on Earth. This would be the sin ancestor or some earlier ancestor to the sin ancestor of the domains of archaea and bacteria. This sin ancestor was most likely an anaerobic thermophile living in a high-temperature hydrothermal environment. Possibly somewhere between the 3.8 billion year mark to the 3.6 billion year mark, we see the emergence of the domains of archaea and bacteria from the sin ancestor. Somewhere around the 3 to the 2.7 billion year mark, we find the emergence of cyanobacteria. Around the 2.5 billion year mark, we begin to find free oxygen gaining a foothold in the Earth's evolving atmosphere as the cyanobacteria produce oxygen as a byproduct of photosynthesis. With the oxygenation of the Earth's atmosphere by the 2.45 billion year mark, the Earth's atmosphere becomes increasingly hostile to its earlier anaerobic life forms. Somewhere very roughly around the 2.3 billion year mark, we might find the first members of the domain Eukarya appearing perhaps in response to the oxygenation of the Earth's atmosphere. The first members of the domain Eukarya would have been single-celled organisms. Fossil evidence from Gabon in Africa points to the possibility of early forms of multicellular life appearing around the 2.1 billion year mark. Around the 600 million year mark, we begin to find the appearance of multicellular animals. By the 541 million year mark, we have the Cambrian explosion and the proliferation of complex animal and plant life. This timeline gives us a rough overview of the emergence and evolution of life on Earth. We will now turn back to a look at the sin ancestor of life on Earth and see what we can discover about its beginnings. As we begin to tackle the nature of the sin ancestor to the domains of archaea and bacteria, let me caution that this is an area of study in which science is still, metaphorically speaking, scratching its head. There are some aspects of the sin ancestor that are agreed upon and some aspects still under debate. This is also an area of science steeped in very complex biochemistry and arcane technical jargon that can be intimidating to the lay person. Be that as it may, we will attempt to forge a path of enlightenment through this complex landscape. Let's start by listing some of the characteristics of the sin ancestor that seem to be largely accepted as plausible based on what we can extrapolate from the more ancient members of the domains of archaea and bacteria. The sin ancestor would have been a single-celled organism with a cell wall. This cell wall enclosed a water-based cytoplasm surrounded by and enclosed within a lipid bilayer membrane. The sin ancestor had a ring-shaped coil of DNA known as a nucleoid which floated freely within the cell's cytoplasm. The sin ancestor's DNA was composed of 4 nucleosides: deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. The genetic code stored in the sin ancestor's DNA was expressed as proteins via single-stranded RNA intermediates such as messenger RNA. The messenger RNA transferred the protein recipes stored in the sin ancestor's DNA to the cell's ribosomes, where the proteins were assembled from amino acids provided by transfer RNA. The sin ancestor's ribosomes were composed of a 50S large subunit and a 30S small subunit. The ribosomally assembled proteins were created using only 20 amino acids out of the approximately 500 amino acids that are known. The fact that the proteins in the cells of all living organisms consist mainly of these 20 essential amino acids points to a common derivation for life on Earth rooted in the sin ancestor. The sin ancestor used ATP, or adenosine triphosphate, as an energy source for metabolic processes. ATP is present in the cytoplasm of all living cells, being an essential energy source for all biochemical reactions necessary for cellular physiological processes. The sin ancestor reproduced by cellular division, also referred to as binary fission. During binary fission, the cell's DNA molecule is duplicated. The two resulting DNA molecules move to the opposite ends of the cell. The cell divides in half, with each new daughter cell getting one of the duplicated DNA molecules. The sin ancestor itself was most likely an anaerobic hyperthermophile. The sin ancestor probably thrived in a sulfur-rich, high-temperature environment, usually associated with geothermal activity such as hot springs, geysers, or deep-sea hydrothermal vents. An optimal temperature range for the sin ancestor might have extended from 45 to 122 degrees Celsius, which is 113 to 252 degrees Fahrenheit. The sin ancestor may have been very similar to the members of the phylum Corachiota, an ancient group of hyperthermophilic, single-celled organisms of the domain Archaea. Two characteristics of hyperthermophilic archaea which may have been present in the sin ancestor are: 1. The presence of the topoisomerase reverse gyrase, an enzyme or globular protein which acts to protect the DNA of the hyperthermophilic organism against thermodegradation or heat damage. 2. Either linked L-lipids found in the cell membrane, which are more stable at high temperatures as opposed to the ester-linked D-lipids found in most bacteria. This quick overview of the characteristics of the sin ancestor gives us some insight into the nature of the sin ancestor. But there still lies before us an even more intriguing question. What came before the sin ancestor? How did life actually emerge on our Earth? This question of the emergence of life ultimately brings us to the quandary of abiogenesis. Abiogenesis is the natural process by which life arises from non-living organic molecules. This seemingly great leap from non-living to living, as we noted earlier, has science scratching its head. What are the basic parameters necessary for non-living organic molecules to become living cells? Life, as we know it on Earth, seems to have 5 major requirements to exist. 1. A source of energy. 2. A type of atom such as carbon that enables complex molecular structures to form. 3. Water, which is critical in supporting the chemical reactions required for biological processes. 4. A place or stable environment favorable to the emergence of life. And 5. The time necessary for life to evolve. As of now, there is no one standard model agreed upon for how life arose on Earth. Various models for the abiogenesis of life on Earth have been put forward. In Chapter 18, we will begin to examine these various models as we continue our quest to understand who we are and where we came from.